Hello students, welcome to Zoology Classroom. In today's class, we are going to learn about osmoregulation in brackish water and marine water animals. So in the previous class, we have seen the osmolarity differences of different water bodies and organisms, tissue fluids or body fluids of organisms. So freshwater osmolarity will be in between 1 to 10 milli osmoles per liter, and marine water's osmolarity will be around 1070 milliosmoles per liter, and osmolarity of blood and other body fluids will be around 300, to be precise, 280 to 295 milliosmoles per kilogram. So these are the values. So according to these values, we can assume that freshwater animals are hypertonic or hyperosmotic to water. Why? Because they are having a more concentration of or more osmotic concentration in their body fluids than the outside water. Whereas the marine animals, they will be having a lower osmolarity concentration or osmotic concentration in their body fluid, so that they are said to be hypotonic to marine water. So in today's class, we are going to discuss about the osmoregulatory mechanisms in marine water and brackish water animals. So in marine water animals, we can find two types of organisms. One is euryhaline and another one is stenohaline. So what are the differences between this euryhaline and stenohaline? Let us see. So euryhaline animals, they can adapt to a wide range of salinities. That means even though there is a wide difference, a big difference in between the salinities of water also, they can survive. Whereas the stenohaline animals, they do not have such ability, so they cannot adapt to a wide range of salinity differences. So only small variations in the salinity waters will be adjusted by those animals. If the salinity changes a lot, the changes in the salinity of waters are big, then the animals, they are not going to survive. So stenohaline animals, they can adapt to only narrow range of salinities, and the euryhaline animals, they can adapt to a wide range of salinities. And these euryhaline animals, they can survive in freshwater, saltwater, or brackish water, any type of water body also okay for these organisms. But the stenohaline animals, it is not like that. Either they will survive in freshwater only, or they will survive only in marine water, but not in both fresh and marine water things. They cannot survive. Why? Because the difference between freshwater and seawater is very big. So freshwater will be having 1 to 10 milliosmoles per liter osmotic concentration, whereas the salt water is having 1070 milliosmoles per liter of osmotic concentration. So there is a difference of around 1060 milliosmoles difference is there. So the stenohaline animals, they cannot live in that type of that much big difference is there. So they cannot survive in that much of wide range of salinity differences. But whereas these organisms, euryhaline organisms, as they can survive in wide range of salinities also. So either they can live in fresh water or salt water, that means marine water or brackish water, also they can survive. And they can live in regularly changing salinities. So even though salinities will be changing regularly, time to time, they will be living them there. So no problem will be faced by them. But these organisms, they can live in fresh water or salt water only. If the salinity of the fresh water changes frequently, they cannot survive. They will die. Either in if they are living in salt waters or marine waters also, if the salinity drops to very drastically, so then also they will be dying. So, and these euryhaline organisms are called as osmoconformers, and stenohaline organisms are called as osmoregulators. And euryhaline organisms will be living in mostly in marine habitats, And ctenohaline organisms, either they will live in marine or habitats or freshwater habitats also. And some examples are also there for these two types of categories. One is molly, the green crab, salmon, eels, and herring fishes are example for euryhaline organisms. And ctenohaline, for ctenohaline organisms, goldfish and hot dog, etc. are examples for this thing. So apart from this thing, euryhaline and stenohaline, another type of thing also is there, that is osmoregulators and osmoconformers. So let us see what are the differences in between these two types of organisms. So osmoregulators, as the name suggests, they can regulate their body fluids depending on the external environment. Whereas the osmoconformers, so they just confront. That means if the outside water salinity is low, their body fluids also will become low. Or if the outside water salinity is high, their body fluids also, they will increase the salinity of their body fluids. So according to the external environment, they will change the concentration or osmotic concentration of their body fluids. But these osmoregulators, they will maintain a constant osmolarity concentration or osmotic concentration in their body fluids, irrespective of the external environment. So they do not bother about this thing, whether it is low to their body fluids or high to their body fluids. They will try to remain the same. They will try to maintain the same osmotic concentration in their body fluids. So these osmoregulators can be marine or freshwater, and osmoconformers, they are only marine organisms. And these osmoregulators, they regulate, as their name suggests, they regulate their internal osmolarity constant. So they keep their internal osmolarity or osmotic concentration constant, independent of the environment. Whereas these things, they will change their body concentrations, body fluid concentrations, or osmotic concentration depending on the outside environment. So in conjunction with the, in correlation with the external environment, they will change their body fluids environment also, or body fluids concentration also. And these osmoregulators, actually osmoregulation can be seen in these osmoregulators controlling the internal osmotic environment. And these osmoconformers, they just simply follow the environment and they do not undergo osmoregulation process. And these osmoregulators, they actively regulate their internal osmolarity. And these osmoconformers, they actively or passively change their internal environment. And these are also called as stenohaline organisms. And these are also called as euryhaline organisms, as we have seen them earlier. And they can survive the osmoregulators, they can survive in a narrow range of salinities only. But these osmoconformers, they can survive in wide range of salinities also. So first of all, let us see the osmoregulation in marine animals. So in marine animals, we can see both osmoconformers and osmoregulators. So osmoconformers, best osmoconformer in the marine organisms is Acmea limatula. This lives in intertidal zones. As it is living in intertidal zones, what is happening? Osmotic concentration or salinity of that intertidal waters will be changing below between 1.5 to 5%. So this is a big change. This is a big difference in the osmolarity concentrations in the water. So such a range of this change in the 1.5 to 5% of change in the salinity of the waters will cause instant death in human beings. So we cannot survive in this range of what change in the osmotic salinity. So but these are able to survive in such type of organisms. So what is the special feature of these intertidal zones? What we have to remember that in intertidal zones, at times freshwater concentration will be high, and at times this marine water concentration will be high. So that in intertidal zones, when the floodwater comes and joins in heavy quantities, then salinity range will go up to this 1.5 range. And whereas during this tufan at times or high tide times, at that time, so more of the seawater comes and joins this intertidal zone. So then the salinity of that region will go up to 5%. So depending on the situations, the salinity changes from 1.5% to 5%. So this is a big difference and this is a big range. So this much of range can be born or can be bear by these Acmia limacula only. So only few organisms can survive in such type of waters. No other animal can survive in such type of waters where there is a big difference in the salinity of these waters. And the sharks and rays are also, they are also osmoconformers. We will discuss about this mechanism that operates in these sharks and rays later in this lecture itself. And another type of organisms, osmoregulators. So most marine organisms, most marine arthropod organisms are examples for these osmoregulators. So out of these thing, Artemia selina, this is called as common name of this thing is brain shrimp. They are one type of prawns only. So they live in salty ponds. They maintain the constant solute concentrations when the water salinity varies from 1 to 30%. So irrespective of the water salinity changes in the external medium, they will maintain a constant body fluid concentration. So whether the body, whether the let's say water concentration is 1% or it may go up to 30% also. So 1 to 30% also, this is a big difference. So in such type of big changes taking place also, the organisms, they will maintain a constant, a stable, the same body fluid concentrations in their body. So due to this osmoregulatory mechanisms only, they are maintaining the stable osmoregulatory mechanisms in their bodies. So when the salts are excess in their bodies, they will excrete, and the main this osmoregulatory organs in these organisms are gills. With the help of these gills only, at times they will excrete salts, and at other times they will absorb salts also. That's why only they can survive when the 1% of salinity is there, and they can survive even the salinity goes up to 30% also. And they live in Great Salt Lake in America. In America, 50 states are there. Out of them, Utah State is one. In Utah State, Great Salt Lake is a lake is there. So the Great Salt Lake water's osmolarity is around 2,500 milliosmoles per liter. So this is very big value when compared to normal sea values. Under normal sea value is around 1,070 only, as we have seen. But this Great Salt Lake water is having 2,500 milliosmoles per liter. That means it is double to the salinity of the seawater. So in such seawaters also, these organisms are able to survive. That means they are the best osmoregulators in the world, we can say. And in other marine invertebrates, how the osmoregulation takes place, let us see. Most marine invertebrates are isoosmotic. Many of these molluscans like squids, they are also isoosmotic. That means they will maintain a same osmolarity concentration in their body fluids as the external water. So as we have seen that external water is having around 1,070 milliosmoles per liter, and they also increase the osmolarity of their body concentrations to up to which is equal to the seawater. So both are equal. So the body fluids are and outside water concentrations are equal. When both the values are same, then we call them as isoosmotic. So by increasing the salt concentrations in their blood and their body fluids, they will maintain this isoosmotic condition. And many of the organisms, many of these invertebrates, they do not have special organs like gills or kidneys, and they are vulnerable to sudden changes. If the salinity of these organisms, salinity of these waters changes suddenly, so big change, if the big changes take place, then these organisms are not going to survive, they will die. So they have adapted to the normal seawater. So if the seawater salinity changes radically or rapidly, then they will not survive in that changed waters. And mostly, what are the problems faced by these marine animals? Let us see that. So as we have already seen, the marine organisms, they are hypotonic to the seawater. Why? Because seawater is having high concentration than the body fluids of the organisms. So the organisms, they face what a problem that they will lose water loss. So they lose the water through their body membrane. So especially the through gill membranes and through some soft skin tissues. So through all these organisms, organs, they will lose water into the seawater from their body fluid. So dehydration is the main problem faced by these organisms. So not only that, through this body tissues and through gills, salt also enters into those organisms. So loss, this what gain of salt and loss of water are the both problems they are going to face in sea waters. So to change these things, what they have chosen, what are the solutions found by them to solve these problems? Let us see. So as they are losing lot of water, what they will do, many of the sea fish, they will drink sea water. So as they are losing the water through their body surface and through the gill surface, so to cover up that lost water, they will drink lot of water. So as they are drinking lot of water, what is happening? Through the salt water they are drinking, through the sea water they are drinking, many of the salts also enter into their body. So water loss is one problem. So to solve that thing, they have chosen this drinking more sea water. So thus they are solving that issue. But another issue is arising here. What is that? Through the drinking water, they are getting lot of salts into their body. So these salts are going to again increase their body fluids than the normal levels, that is 300 milliosmoles per liter. So salt gain is the problem. So to solve this salt gain, they have, the gills have again this what secretory cells are chloride cells. So through the chloride cells of these gills, they will be secreting the excess salts into the seawater. So the same thing happens in reverse manner in the freshwater animals. So freshwater animals, they are gaining water through the body surface and gills, and so that what they are doing, they are urinating very dilute urine and ammonia. And to gain the lost salts, they are absorbing the salts from the waters through the chloride cells of the gills. So here the gills are also having chloride cells in marine animals. The chloride cells of these marine animals, they are excreting salts into the sea waters. So reverse occurs, reverse action will be taking place in marine animals. So we can see the all those why we have discussed in this diagram. So the kidneys of these organisms, so they will release many salts into this urine also. So urine will be concentrated urine and less in quantity. So very concentrated urine will be secreted, into which many excess salts will be excreted into this thing. And here gills are having chloride cells. So these chloride cells are also what they will be doing. They will be excreting sodium chloride and other minerals, excess salts through these chloride cells. And what is the problem? Through this total body surface and through the gills, water will be going out of the body. So to compensate the lost water, they are drinking lot of water. So through the drinking water, they will be gaining the lost water. And through food also, in food organisms, the body fluids will be there. So the body fluids of the feed organisms are also, they are going to gain the water. So in this way, they are gaining the water in the form of food and sea water, drinking sea water. So by doing so, they are solving the water loss problem, but they are gaining lot of salts. So to lose those salts, they are what they are doing. They are excreting those excess salts through the chloride cells which are present on the gills, and through this excretory system also, they are creating a very concentrated, highly concentrated urine, into which many excess salts will be secreted into that urine. So this is how the marine fishes, Osteichthyes fishes. These are Osteichthyes fishes. So marine Osteichthyes fishes solve those two problems. And in sea waters, we also find cartilaginous fishes, shark fishes and ray fishes. So in shark fishes and ray fishes, these are not osmoregulators, these are osmoconformers. Okay, shark fishes and ray fishes, these are not osmoregulators, these are osmoconformers. That means they will maintain almost isoosmotic body fluids in their bodies. So outside the seawater is having 1070 milliosmoles per liter osmotic concentration. The body fluids of the shark fishes, ray fishes, and many cartilaginous fishes are chondrichthyes fishes. So all these fishes also, they also will be having around 1070 milliosmoles per liter concentration in their blood and in their tissues and in their cytoplasms also. So in the all these body fluids, they will be storing many salts and urea. Urea also will be stored by these organisms to just to increase the concentration of their body fluids. So if the urea is going to stay in the body for a long period of time, that urea is going to damage the tissues. So to counter this urea bad effects, they are also storing TMAO, trimethylamine oxide. So trimethylamine oxide is a special chemical which saves the animals from the bad effects of urea. So that's why this urea and other salts are helping the animal to increase their body concentrations, which will be equal to the seawater concentration. But in doing so, the urea is going to damage the tissues and organs. So to prevent that, they are also storing another TMAO chemical, which prevents the bad effects of the urea and other salts on this animal. So, and these organisms, they will not drink. So, other ostrich fishes, these fishes, they will drink water to uh, cope up to cover up the water loss. But these organisms, as they are not losing water, why? Because they are isoosmotic. So, if some amount of water goes out of their body, the same amount of water enters into that body, so that no water loss problem will be there in this in this chondrichthyes fishes or cartilaginous fishes. Why? Because they are maintaining a isoosmotic concentration in their body fluids. So that's why these are called as osmoconformers. So as they are not losing any water, they what they will do, they will not be drinking any water. And kidneys also, what they will do, they will conserve urea. Most urea will not be sent out of the kidneys. They will be again reabsorbed into the blood just to increase the concentration of the body fluids. So through the urine only, sodium chloride and some amount of salts only will be going, and very less quantity of urine will be produced by these osmoconformers or cartilaginous fishes which live in seawaters. And osmoregulation in other marine animals. So in birds, some birds, there is a special bird called, whose scientific name is Macronectus giganticus. So in this Macronectus giganticus, not only in this Macronectus giganticus, all remaining seabirds also, in seabirds also, they will be having a pair of special glands on their nostril region. They are called as nostril glands. So these are the nostril glands. So here one pair of nostril glands are there. So inside these nostril glands, what they will be having, they will be having some canals, nasal canals, and these nasal canals, they are highly they are supplied with high vascularization, or many blood vessels will be there. So through these blood vessels, so here you can see that, so this is the blood vessel, this is the wall of this duct, nasal duct, this is the lumen of the duct, and the lumen will be open to the exterior through a pair of holes, nostrils. So into these nostrils, these lumens, the pipe lumens, so the lumen will be open into this nostril pipe. So from this blood, what is happening? They will be secreting excess salts. Excess salts will be secreted into this central canal of these ducts. So lumen of this duct. So the salts that enter into this lumen, finally they will be secreted through these holes on which are present on the nostrils. So in this way, by having this special secretory nasal glands, these nasal glands, they will be filtering all the excess salts in the blood, and excess salts will be taken into this lumen of these nasal glands. So through this nasal glands ducts, the excess salts will be secreted in the form of tears. So like this, this type of special nasal glands, this type of special arrangement is helping these organisms to lose the excess salts. And osmoregulation in reptiles, in these reptiles, So the reptiles are also, they are osmoregulators. The body fluid will be maintained at 300 milliosmoles. Normal mammals body fluid concentration also will be maintained in these reptiles, marine reptiles also. So they also will face the same problem. So what is that problem? So they will be losing water. So to prevent that water, they will be drinking seawater, and salt glands are also present on the eyes of these eye glands of these organisms. We know that in crocodiles also they will be secreting tears. The tears actually are nothing but they are losing the excess salts from their blood. So the excess salt which is present in their blood will be collected by the salt glands which are present on the eyes, and through this eyes they will be excreting that sodium chloride or excess salts. And the urine generated by these organisms will be isoosmotic. That means that will be equal to the body fluids. That means around 300 milliosmoles will be urine will be there. So like this marine reptiles and birds are also, they are also osmoregulators. They maintain around this 300 milliosmoles concentration in their body fluids. Coming to marine mammals, so marine mammals are also, they are also osmoregulators, not osmoconformers. So they also maintain around 300 milliosmoles per liter of osmotic concentration in them. So these organisms, they are having these Henle loops like we are having. So marine mammals like these dolphins and dugongs and all those things, whales, every marine mammals are also having the kidneys just like us. So they also will be having this what Henle loops in them, so that these Henle loops, they will be helping them to reabsorb lot of water. So they will be excreting a highly concentrated urine. So, but these organisms, these fishes, these what reptiles and birds, they are excreting isoosmotic urine, but marine mammals, they are excreting hyperosmotic urine. So the body fluids concentration is 300, but urine concentration will be around 1200 milliosmoles per liter. So they are excreting more concentrated urine than these marine reptiles and birds. That is the difference. So, and they also osmoregulation will be done in these organisms, but these organisms, these marine mammals, they will not drink seawater, so that they are trying to conserve more water. That's why only as they are not drinking seawater, they are excreting very concentrated urine to conserve the water. So this is how these marine mammals and remaining all organisms are going to regulate their osmotic concentrations or body fluid concentrations. Then next, coming to the brackish water animals, how they are going to regulate their osmotic concentrations in their body fluids. So these brackish waters have more salinity than fresh waters. More, not in a type mistake. More salinity than fresh waters and less salinity than seawater. So they are having an intermediate. They are intermediate to this fresh waters and sea waters. So it's sometimes they are very close to fresh water and sometimes they are very close to sea water. So these conditions will be changing. These conditions will be changing depending on time and space. That means at some places one type of salinity will be there, at other places other type of salinity can be seen, and it is single place also over a time. In some seasons salinity will be like so at some other times salinity will be different than previous time. So likewise like this, the salinity levels in the same place will be changing over time also. And these brackish water conditions also will be seen in estuaries and intertidal zones. So in all these three things, brackish waters, estuaries, and intertidal zones, in these three areas, the same conditions will be prevailing. And the animals which are living in these organisms in brackish waters or in estuaries and in, or intertidal zones, in all these waters, the animals are definitely, they are urihalin. Why? Because they have to adjust to a wide range of differences in the salinity levels. So, and they can behave both like freshwater animals and marine water animals depending on the salinity levels. So when the salinity drops to minimum level in these waters, then they will behave like freshwater animals. That means they will lose, they will excrete a very dilute urine and they will absorb salt from the water. When the salinity of the water goes near to marine water levels, then what happens? They will behave like marine animals. Like what? They will be drinking water and they will be excreting concentrated urine and they will be secreting salts through the gills. So likewise, when the salinity is low, they behave like freshwater animals, and the salinity is high, they behave like marine water animals. So the same chloride cells, at sometimes they will be absorbing salts from water, at some other times they will be excreting salts from their body fluids. So both ways, the organisms, both ways, their systems are going to work. And very few animals only have this capacity, so that in these animals we cannot find a wide range of animals in these organisms. Some exclusive animals, so that means only some animals can be seen in brackish waters or estuaries or intertidal zones only. Those organisms are called as exclusive animals. And some other animals are called as migratory animals, like salmon fish and eel fishes. The salmon and eel fishes, usually they will be living in sea waters, but for breeding purpose they come to fresh water. So at some other some period of time they will spend in their life in marine or marine habitats, and some other part of their lifetime they will come to fresh water. So both type of situations they will be experiencing in their bodies. So these type of special migratory fishes are also example for they will be having this type of capacity. And in these organisms, so Poecilia, Physialia spinops. This is called as black molly or common molly. So the lower picture is common molly, and upper picture is the black molly. So these molly fishes, they can live either in brackish waters or sea waters and fresh waters. So in any type of water body also they can survive. That means they will behave accordingly to the water in which they are living. And another thing, green crab, Carcinus minas, and eel fishes. So these eel fishes and Carcinus minas are also, they also can live in waters in which wide range of differences can take place. So these eel fishes, they will live in sea waters, and for breeding purposes, they come to fresh waters. So when they are living in sea waters, they behave like marine organisms. And when they come to rivers, they behave like freshwater organisms. Like this Carcinus minas or green crab also has the capacity, same capacity. And salmon fish also, we can see in this diagram very clearly. Both these two pictures are of the salmon fish only. So when it is living in freshwater organism, so through body surface, it is gaining water. So this blue color is gaining water, and the salts are also going to enter into this body, into its body. So to prevent this excess water problem, what they are doing, they are excreting dilute urine, and this what gills are also having. Those gills, what they will be doing, they will be absorbing salts which are going to lose through the urine. So and the same thing occurs in reverse manner when the salmon fish enters into this seawater. So in sea waters, what they will be doing, they will be losing the water from their body surface. So to cope up that, it will be drinking water. Much amount of water will be drank by this organism. And here this gills, the chloride cells which are present on the gills, they are excreting this excess salts. And here the urine, concentrated urine will be excreted when it is living in salt water. So this is how these osmo conformers are urihalline organisms. So these urihalline organisms, they will change according to this external environment. So, so with this, we have finished today's topic. Let us meet with another topic in next class. Till then.